we just introduce with computational pathology, and then we would introduce a little bit of our work in AstraZeneca, which we did. So not only my work, but also some of the work from the colleagues, where we use deep learning and computational pathology. So what is computational pathology? So it basically is a cross-functional basically workflow where we analyze cancer images and help to make some decisions for therapies. Our major focus is on images. So what we do is so basically it starts from diagnosis. When a patient is diagnosed with cancer, then the sample is acquired, it's cut into blocks and stained, digitized. So we have gigapixel images most of the times of tissues, which we need to analyze automatically with deep learning. And on that, we do different kinds of analysis, like on regions, on cell level. And in the end, we have to integrate all the data with not only image features. So these basically will provide us some features of cancer in general, which we would need to link with survival data. And there are also different sources like genomics, which we can all integrate. And then in the end, do data mining on them and provide some insights on cancer biology or basically predictive biomarkers or something like this. So in AstraZeneca, we focus mostly on this part, at least our group, and we do not focus on the sample acquisition and digitization. This happens in the labs. And most work we talk about today is focused on these parts. Like you already might imagine that all of our work is on images. So I would just introduce a little bit of different types of images, what we get. So we have different kinds of stains and scanning technologies in histopathology. So this on the left-hand side is the HND image, which is basically this pinkish purplish, which basically is the most basic stain used to check the morphology of cancer in general, basically growth patterns and everything. So pathologists really prefer this kind of a stain to check the tissue sample. The second type of stain is the immunohistochemistry stain, which targets certain kind of regions or cells. So in this case, you see these brown regions. These are all epithelia, and this is a cytokeratin marker in this case, which stains all kind of epithelia. This is only one kind, and we have different kinds of IHC, basically, which will target different kinds of cells. And I would show some examples later on in this slide, where we analyze the IHC images. And the last part is multiplexing, where we can have basically multiplex of different markers. So here in green, you see cytokeratin, which you see here, for example, in brown. You also see DAPI channel, which is basically nuclei, which you see on the left-hand side, basically this purple. And you see some red here, which is some immune cell populations, like CD8 or something. So these are different kinds of images we have, and which all provide different granularities of analysis or opportunities for us in this case. So when we talk about analysis, we generally go towards deep learning. They are the latest and greatest methods to analyze these kind of data. And traditionally, we also fall back on fully supervised deep learning, where we ask an expert to basically draw regions of interest. For example, these in pink or yellow, which whatever we need to analyze, and then just feed this to a deep learning network to basically analyze these kind of regions automatically. So this is one of the simplest way to do this, but very time consuming. As you can imagine, we would need an army of pathologists and experts to draw these kind of regions on different kinds of images and cells. So this is basically our kind of baseline where we come from, and we want to improve on that, right? So we want to reduce this dependency on pathologist annotations, and that's why we try to develop new methods here in AstraZeneca to basically reduce this demand of annotations, as well as at the same time make the methods faster and have a more robust analysis. So I just collected some applications which we have in AstraZeneca, which goes from left to right in terms of deep learning novelty. So on the left-hand side, there are some applications which are fully supervised learning. So you have, for example, here, cytokeratin detection, which is basically a simple method. Also, but here, if you see, I don't know if it is visible, you have these dots, basically these TILs, so tumor infiltrating lymphocytes, being detected on HND images, and these are both are examples of fully supervised learning. Here in the middle, we have some semi-supervised methods where we have a very few annotations from pathologists, and we try to obtain automatically or synthesize some annotations automatically from other domains. I would talk about this work in detail in this talk. And last but not the least, we have some fully automated unsupervised methods where we take the histopathology image as an input and we try to train end-to-end with survival data to basically produce predictive biomarkers and to capture long survivors versus short survivors. This is also a topic for today, and we will also give some details on this work. Any questions till now before I jump into methods and details? If not, I will start with the first work from Natalie. I was a co-presenter in this work. So this is basically segmentation of prostate glands in HND images. Again, the focus is on how do we reduce the requirement for manual annotations in this kind of an experiment. Just to introduce the problem, so the glands in prostate look like these, and this is what we need to analyze automatically. The nice thing about the cytokeratin marker, the cytokeratin 18 or P63 marker, is it automatically stains the glands, prostate glands, and then it's very straightforward to do some basic image processing, like just detecting brown and then getting the regions out. Now the challenge is how do we use information from this stain and automatically derive annotations and results on NHND stain. And this is what we would do. So if we have consecutive sections, so if we have consecutive sections on HND and CK18, then it enables automatic transfer basically of this information from brown to this layer. And this is what we would do in this work. So we would automatically co-register these images so that these sections are close by. And then we automatically detect these regions on HND. So just an example of how this looks like. So this is HND stain, this is cytokeratin stain. What you can see here, if you focus on this region, is even if these are serial sections, the information is not exactly the same. So you see here, these are two glands here, which is, I think, this is going some kind of change in the category. Sorry. But here the gland is different. So even if we use directly this information, this is not supremely accurate and it has some noise. And the challenge becomes how do we deal with noise in this case? And yeah, so here is also, you see here, so this in yellow, this is when we overlay the information from cytokeratin on HND. So this is the annotation we basically obtain. This is noisy level, weak level, but this is still, we attempt to train a simple unit, I'm sure. So this unit architecture is a very, very common architecture, like one of the traditional architectures which we use for semantic segmentation in deep learning. So basically, we just try and use this noisy level to predict. What we see, even if the input is noisy, you get the prediction which is much smoother, which is nice to have, at least in the first attempt. Again, here we see some instances. So here you see sometimes the information from cytokeratin, which is in yellow, is much nice, but sometimes it's way off the mark. So for example, here it's like way off the mark and it's very, very noisy. But when we apply back the trained model, we see that these networks are very robust, sometimes robust to this kind of noise and gives you very accurate results. What we see still here is, if you see these are two glands here, and there's a separation in between, but this kind of noisy levels does not separate like singular glands. So here, if you see 1, 2, 3, 4, these are all separate, but you get only one prediction. So this is the next challenge for us. How do you separate these glands into individual glands?
Sorry,
and
Ansh,
here, yeah, please.
can I ask? In the image above, what's the problem is what the things are not aligned in the original image, or
exactly. So this is aligned. So this is the best alignment we can get with the serial sectioning of cytokeratin 18 and HND. So you segment cytokeratin 18, which is basically yellow, and you overlay the information on this HND image. And this is the annotation we use then for training the model directly on HND. So this is basically misalignment in this case.
And so you have specific laws that allows for shifts, or?
In this case, no. And I will come to that, how we deal with this case. But what this just shows is, even with this kind of noise, the network is able to learn something. And it's not exactly, it does make sense still. So you see the output is very nice segmentation, even though it does not segment individual glands in this case. And this is the next problem we'll try to solve. So yeah, what Nathalie did was basically, in the end, we have to fall back to manual annotations. So she made 73 like polygon annotations around these glands, like here, so like this. I don't know if it's visible. I think it's very faint. Maybe here, for example, you can see, so this kind of annotations like singular, and you see there's very little space in between. And to fix this, we use basically high weights on borders so that you get separate glands in this case. And we just did iterative refinement again. So we had a model which was pre-trained. You do this iterative refinement with high weights on the borders of glands, and then we get a very nice segmentation where we get individual glands with a die score of nearly 0.95. So this is basically coming from an imperfect segmentation, which is on the left-hand side, to individual glandular segmentation, which is in the middle. You see the input on the right-hand side. So this is basically done iteratively, and you get this result for segmentation. Any questions on this? This is a simple method, but
hey, Ansh. Yes, Peter, also from TUM, actually, from Munich. I have a question. How do you align these two modalities? Is this done manually, like the IHC image to the H&E image, or is there also a method for that?
There are co-registration methods, which I did not touch upon, but in the first step, this is basically you try to align these two stains together. So this is co-registered. I think I am not entirely sure which method was used in the end, but this is also rule-based co-registration. So you have some landmarks, and you try to align the landmarks together to get a registration. These two stains.
Thanks.
Any other question on this method? Okay, so just to summarize, we had the first proof of concept that we can have this automatic generation of ground truth images from consecutive sections, and this makes sense. This also works out very nicely in the end, and then we have this novel boosting approach. So when we do this iteratively, not only once, but like we train on the predictions of the model, and this helps to improve segmentation over time. And we will try to implement this, apply this on further cancer subtypes, also different indications, and to automate the scoring methods in the future. The next work which I would touch upon is also building on basically this work, where we want to use information from other domains. So that's why I use this word domain adaptation. So in this case, also domains will be two different modalities of images, and we want to segment, in this case, PDL1 images for epithelial regions. Before that, I would just give some context on why we are doing this. So we are doing this on lung cancer samples, also advanced, stage 4, which has a median survival time of only 6 months. And if the treatment is standard of care, so for example, chemoradiotherapy, then the 5-year chance of survival is only 1%. And in this domain, so basically in this setting, AstraZeneca have a drug called durvalumab, which is rather an antibody. So we have a drug which is showing very encouraging signs. And here, the PDL1 biomarker status is predictive for treatment. So it's natural to see if we can automate getting the status from histopathology images and see if that makes sense or not. So this is a little bit of background. How do we establish PDL1 status is very straightforward. So it's defined by a tumor cell score, which is nothing but we check so the amount of cells which are PDL1 positive divided by the total number of cells which are PDL1 positive plus PDL1 negative. So it's basically the ratio of positive PDL1 tumor cells on the whole slide image in this case. So, and this is how PDL1 IHC images look like. So you have, so brown means high PDL1 and non-brown means low PDL1. And this is the challenge that we have to automate this tumor cell scoring in this kind of images. And what I was mentioning, that PDL1 status helps in identifying patients. So you see in green here are the patients which have high PDL1 status defined by a TC score greater than 25. And in the blue arm, you have lower survival probability. And these are patients with low PDL1 status. So at 50% probability, so somewhere here, you have, I think, 20 months of overall survival with in PDL1 positive arm versus around 10 months in PDL1 negative arm. So in short, if we treat the patient with durvalumab, then you have 10 months of survival benefit over chemotherapy in this case. And this is what we want to automate now. Why do we use deep learning? Because you can say that it's we can use a Brown detector on tumor cells and get the score done, but PDL1 is very difficult. As you can see, so this area here is a true PDL1 positive region. So all the cells here are PDL1 positive, but at its side, you have this area which is PDL1 negative, but it does not only stain tumor cells, but you see also in the stromal regions that you have some positive immune cells. You have blur sometimes, so you don't even see how the tissue histology is underlying, and you also have unspecific stain. So to have rule-based analysis on this is very, very difficult. We tried this in the past, like in pre-deep learning era, but it was very difficult, and yeah. So this is why we chose deep learning. And yeah, so, and again, the same challenge comes. We can, of course, manually annotate everything, like you would encircle regions of PDL1 positive and PDL1 negative, and that would also work, but we want to minimize this effort, and that's why we want to use, again, data from other domains. In this case also, the data will be pan-CK. But the challenge here is you want to also identify, so you want to separate out tumor cell positive regions, which are basically highly brown, and tumor cell negative regions, which are unstained for brown. So what we do is basically synthesize a cytokeratin image, which you see in yellow, into two images, one which is unstained for PDL1 and one which is stained for PDL1. This synthesis is done by a GAN, so I would come to that. So basically a deep learning network which synthesizes these. And in this way, we can collect a lot of annotations, because from pan-CK, as from the work from Natalie, you can automatically get basically epithelia area. And these annotations here, which you see, this is the mask. So in white, you have epithelia, in black, you have non-epithelia. This is generated automatically in this case. In addition to the synthetic data, you have also real annotations from pathologists, very few annotations just to basically fine-tune the network in the end and to have good performance. So yeah, coming to the architecture, I would not go into too much details, but this is basically a schematic on how this cycle can work. If there are questions, I can take them. So there are basically two networks, one to convert PAN-CK image, which is yellow image, into PDL1 image with brown staining. And there's another network which converts a PDL1 image to PAN-CK. And this works in a cycle to balance out. And in the end, you have one segmenter, basically segmentation network for PDL1, and one segmentation network for cytogenet, so PAN-CK in this case. This is a GAN, so generative adversarial network. I would not go into too much detail. I can take questions, of course. This is basically a new way of adversarial learning, which helps in better synthesis of images. So this synthesis can be done also by other methods, but you get blurring in those cases. But with GANs, you get very sharp reconstructions, as you see here in this case. So this is basically what we did. And this domain adaptation, so basically conversion from PAN-CK to PDL1 and vice versa, plus the segmentation, basically happens in conjunction. And this is a joint network which we can train end to end in this case. And yeah, so basically when we input, once the network is trained and we input an image, we get different regions. So in red, tumor cells negatively stained, so basically unstained for PDL1. And in blue, we have tumor cells which are stained for PDL1. And from this segmentation, we can compute the TC score. Basically the sum of tumor cell positive by tumor cell positive plus negative. And what we did here was also validation of this score against pathologist scoring. So we had 700 slides where a pathologist scored these slides for PDL1, and we correlated that. So on x-axis, for example, you see the scoring from pathologist from 0 to 100. And on y-axis, we have the estimated one from the machine. And we see there is a very nice correlation across the whole range, where we have the concordance of 0.93. So it's really, really nicely concordant. On the left-hand side, we show basically the results of segmentation. So the 2+ segmentation is segmentation of all epithelia from everything else. And the 3+ segmentation is segmentation of also the classification, in this case, of epithelia into tumor cell positively stained for PDL1 and tumor cells which are unstained for PDL1. And both of these show best results with DASCAN, which we can see in orange bars here. Also, we did the experiments against fully supervised networks. So here you have what you call SegNet, which is only trained on manual annotations from pathologists, against this proposed semi-supervised method. And we also see that this method always works best for increasing amount of available annotations. So on the left-hand side, in first case, we have only 22 slides, which are annotated by pathologists, and we increase that in steps to see basically the effect of having this semi-supervised learning. So we see the most effect when we have the least annotated data. So you see the difference from 80 to 85. But when we have substantial annotations, then the performance of the two networks are similar. So it just shows this network. So this new method does leverage from other domains, and this also helps learning and to make the network more robust in this case. Any questions on this before I move to the next one? I know it's slightly complicated, but I can take any questions. Not.
So GANs are there problems of stability to fit models like this, or?
GANs, yeah, they are very tricky to train, and but the cycle GAN setup. So when you have the kind of a cycle where you have basically this PAN-CK to PDL1 and back to PAN-CK, this kind of a framework is more stable than if you only try to convert PAN-CK to PDL1. at least in our experience. So the cyclic setup is, one of the most, in my experience, one of the most stable setups what I've experienced for GAN training.
And so if I got it right, you can do this without actual paired images and run and fit the entire cycle.
Yeah, so with this network, you don't even need paired images because you're synthesizing separately. So this is one of the advantages, like I mentioned, from the previous work, where you need always paired images. In this case, you do not even need paired images. So we had from 2 different cohorts, the PDL1 and the PanCK images. So yeah, so this limitation what we had in the previous work is elevated in this work as well. So we can have any source, any 2 markers, not only PDL1, but any other marker, and we can have this network running in no time.
And did you apply this for more type of stainings now, or?
We did internally, but I cannot show this because that is still unpublished. So this can be done for all epithelia markers because PanCK is epithelia. So if you have any marker which is staining epithelia, not only PDL1, but for example HER2, you can do this kind of synthesis and task on training in that case.
Okay.
Cool. The third one is basically the most novel application, which I think Armin from our group drove in that time, where we tried to predict end-to-end patient outcomes from patient outcomes using deep learning. So there were no pathologist annotations, only survival data and images available to us in this case. So yeah, okay. So Armin did this training on tissue microarrays. So we had tissue microarrays from gastric cancer. So these are like these small pieces cut from the whole tissue, the most interesting regions. And there were 248 patient data in this case, and there were 5 markers. So CD8, which is cytotoxic T cells, CD20 for B cells, CD68 macrophages, proliferation marker, and like CK epithelia cells. So what Armin did is, so in a survival training network, we directly sample from images, so the small patches where we train a network. Each patch here is associated with a time and event. Basically, time is your overall survival, and event is if the patient is still alive or censored or dead. And every patch, so use patch like small one from here and yeah, input into the network. So this is patch from all of the cores, and yeah, and in the end we train a simple classification network. In this case, we used Google InceptionNet, but the loss was Cox loss. So this loss here is derived from Cox proportional hazard model, where this term here XIV is your risk, and this here is a normalizer. So basically, this takes this normalizes the risk for the patch across the whole cohort. And so for each patch, we will have one risk value attached to it. And this is learned automatically using survival information from images. Just to visualize how the output looks like. So we train the model, we apply it on the whole core. And this is how the output looks like. So you have the heat map on basically high risk and low risk regions. So this is just an example of CD20. So what this shows is basically if you have more number of CD20 cells, you have low risk. That means the patient will have high chance of survival. This is all learned from the network using survival data in this case. Also, another example, so if you do not have any CD20 in the image, we see this is associated with higher risk, which is indicated in red here in this case. This is just to visualize what the network is learning. Yeah, and Also, this is seen in the research, basically in other research, that B cells or CD20 are associated with favorable prognosis in gastric cancer. So this is what we found in literature, just to have this correct interpretation of these risk maps, so that we are sure that the network is not learning anything which is artifact, but it is learning some real biology in this case. And this is also shown in this example, so that you have this TLS structure, which is favorable for survival. And yeah, also this high density of CD8 cells, all these brown cells, which gives you favorable prognosis in this case. Now the challenge comes, so we have a big TMA core, and we can have a heterogeneity, heterogeneity in the risks. So you have red regions, basically high risk regions, like in this pink, to slightly low and to favorable regions. How do we aggregate this information to a single value, which we can plug into stratification? And in this case, we checked the distribution of all the risks, and it was normally distributed. So basically, we took the median of this distribution to basically assign every TMA core one risk value. And using this risk value, we stratified the whole cohort, again, into two branches, equally distributed. And we can see the ones from with higher risk, the one with lower risk have higher chances of survival, and vice versa in this case. Arvin also did the Cox regression with other covariables like age, gender, MMR, and BMI. And we saw that these survival learned features automatically from the network are the most significant in this case, with the highly significant p-values as well. One more thing I would like to maybe note point is we are not limited to have only this feature, but we can plug in any kind of genomic feature or nanostring feature to this layer and have a combined learning of image plus genomic features in this case to have a end-to-end learning. So maybe this is interesting for the community in general to basically how do we do multi-omics analysis in this kind of a setting. Any questions to this work? And then I will move to the last work, which is also an extension to this one. No, nothing. Okay.
That was okay. That was clear.
Okay. The last one I would just do, so this is basically the work which we published recently in Transactions of Medical Imaging, and this is the extension of Dasgan work, which I showed previously in this talk, where we tried to basically use region-focused survival learning. So what Armin did was basically unfocused. So we took the whole sample, created patches out of it, and fed it to the network. We tried the same approach on a clinical trial sample, we failed. And then we tried to solve that problem, and that brought us to this region-focused survival learning. In this case, the difference from the previous work is we, like the name suggests, we want to focus on certain regions. In this case, this is the same data set, the PDL1 data set which we were working on. So we use the Dasgan to segment out epithelia. In this case, we patch uniformly from a grid, assign each patch to a time and an event, and then train a network to predict risk scores. So the only difference from the previous work is basically the sampling, that in a clinical trials, because we have a mixture of biopsies and resections, we need to balance the sampling, and we sample only from epithelia. And what we see is when we apply this model from part B to part C, we see if you have tumor cells which are brown, you have low risk and high risk otherwise. And we aggregate the same way. So in this case, this distribution was no longer normal, and we tried to split it at a quantile here. So I don't remember exactly if it was median, but I think it was. And yeah, so before this red line, you have everything as low risk, and on the right hand side, everything is high risk. So it's very similar pipeline, just we focus on a certain kind of region. And this is done on PDL1 images, in this case, on a clinical trial. Now coming to different kinds of stratification. So on top left, so this pathologist scoring is what we did in work number 2, which I showed today. So we computed the TC score and we tried to stratify. So this is this part number A is when the pathologist scores a sample. So we get still significant results. So p-value of almost 0.01. But we do better when we apply the same rule. So TC score greater than 25 within image analysis based TC scoring. So we see, like we saw in the correlation plots, that we get similar results. So we can confidently say with this result that we can automate this scoring process and it would give comparable results to a pathologist in terms of TC scoring. In terms of survival learning, we, like I mentioned, we did this non-focused. So basically we sampled from the whole tissue, the patches, and we saw that we were not significant at all versus region-focused survival learning, in which we are highly significant again. In stratification. Also, this is just additional analysis when we tried to see TC scoring from the pathologist versus automated. So so this line here basically is your 25% cutoff. So the only non-concordant cases are these ones. Out of 704, I think we have a few cases which are non-concordant, which would change status from pathologist high to automated low and vice versa. So this quadrant, top left and bottom right quadrant, which is, this is, And then just to see how the risk scores relate to pathologist TC scores, we plotted this graph, and we see on the top left corner, we have high risk associated with low PDL1 status and low risk associated with high PDL1 status, which is consistent with biology, which we expect. And I guess this was my last slide. And just to conclude the work from Armin and from us, we established an end-to-end pipeline where we just take the images and learn survival from it. This is fully automated features and parameter learning, so there is no intervention of pathologists. This is cross-validated, so I didn't mention in the talk, but this was cross-validated, so this is already statistically significant. And like we showed also with PDL1, this is easily adaptable, where we just need to plug in different markers and we can check if this makes sense or not. What we still need to do is, for different markers, we need to check in depth if it makes biological sense. For CD20, it did make, but it is possible that for other markers, it does not. And this is what we need to check in the future. In this work, all the patches are independent. So we did not take any kind of consideration of space. So every patch was individual. So, and it might make sense that we take basically neighborhoods of patches and this information into consideration. And that also might give us more information in the future if we train with, for example, some kind of graph structures, this kind of analysis. Also, like I mentioned, that we can integrate many different omics with this kind of a methodology, like gene expression, also location subtyping. It's just a variable plugged into the last layer before final fully connection in the Google net. And we need to, of course, validate this on an independent trial. So this was cross-validated, like I mentioned, on the same trial. But if we have this done on an independent trial, then that gives us new level of confidence that this works very nicely. And additionally, not only time, we can also try to predict treatment response with this kind of an end-to-end method. I think I am in time. If there are any questions, I'm happy to take them now. and thank you for your attention.